Hello my dear students, a very good afternoon to all, and I welcome you all to the most interesting and interactive live session. This is Vasavi Ma'am, your biology educator for Foundation and CBSE. Yes, students, so let's have a look on this. In our institute Neet World, we have already started the foundation classes for the classes 6th to 10th children. Okay, and here we are going to offer you an exclusive courses based on the subjects like physics, chemistry, mathematics and biology, and we are going to cover each and every chapters for both the sessions like CBSE as well as SSE. And in our institute Neet World, we are going to cover the chapters which are very important for NEET and JEE. And next, after completing each and every session, we are also going to have a special doubt clearing session, children. So by that you can have the clear concept and clear idea on the particular chapter. And next, we are going to make each and every class in an interesting way by creating some animated videos to make the concept clear, children. So this is our institute Neet World, which is located in Narayanguda, Hyderabad. Yes. Yes, here comes an most important news for all the students of the class 6th to 10th standard students. Yes, and it is all about the scholarship test, children. Neet World Star 2025, it is all about the scholarship test, and all the students from the class 6th to 10th are eligible for this particular scholarship test, and the dates of the scholarship test has been extended, children. Okay. So you have to, all the thing you have to do is you have to download the app called Neet World. So by this app you are going to get the notification of the particular scholarship test examination. So by that you can join our institute and you can happily give the scholarship test examination, children. And when it comes to the benefits of this particular scholarship test, you are going to get 100% of the scholarship on NEET and JEE foundation, and next special cash awards are also being provided to the top performers, and next we are also going to give the personalized guidance and career counseling is also being provided from the experts, children. So coming towards the mode of examination, it is both in the online and offline mode, children. So it is as per your preference to attempt this particular scholarship test examination. It is just to test your talent, children. So please, so what are you waiting for? So please just join our institute Neet World and download the app Neet World, and you can attend the examination either in online or offline mode. And by writing the scholarship test examination, you can evaluate your potential, you can boost your confidence, and next you can access the quality coaching. This is all about the Neet World, your step to medicine. Yes. So children, all the 8th standard students, I think all of you have done well with your examinations, and now in this new academic year, so in this particular session we are going to start the first chapter, that is Cell, the fundamental unit of life. It is the first chapter of your 9th CBSE syllabus, children. So it is a very, very important chapter when it in the examination point of view. Yes. So in this particular chapter, yes, in this chapter, if we see the weightage of this chapter, it is nearly 25 to 30%. Children, totally you are going to have 6 chapters in your 9th biology CBSE syllabus. Okay, and we have already discussed and we have already done the session based on the previous paper analysis, based on the sample paper analysis, based on the weightage and syllabus coverage. We have done the session on this particular study, so you can go through that and you can get a better idea on this syllabus, children. So in this chapter, Cell, the fundamental unit of life, it is going to occupy the major weightage. Okay, it is a very, very, very important chapter. All of you have to be stay tuned until the session gets end. Okay, and it is a very important chapter where you can get the maximum number of questions. So you can cover the 25 to 30% of the weightage in this chapter. So please stay tuned and okay, and next try to understand all the concepts in a clear way. Yes. So what we learn in this chapter? Clear? So what we are going to learn in this chapter? Yes, in this chapter we are going to cover some important contents like, first we are going to learn about the general introduction of the cell. Yes, and next we are going to discuss about the discoveries of the cell. So in this topic we are going to study about some important scientists and their theories relevant to the discoveries of cells. And next we are going to study about the microscopes. Okay, these are actually the instruments which are used to visualize, which are used to see the cells which are invisible to our naked eyes. So in these microscopes subtopic, we are going to learn about the two different types of microscopes. Okay, one is electron microscope and one more is compound microscope. Okay, these two are the types of microscopes which we are going to learn in this chapter. And next we are going to have a brief look on the prokaryotic cells and the eukaryotic cells. This is also very important topic, children. Okay, so here you need to memorize the diagram as well as its functions. Yes, and next basic idea about the cell wall, and next one is plasma membrane. Okay, so in these entire contents you need to focus mostly on this plasma membrane structure, children. Okay, so from this plasma membrane structure only you are going to get the type of questions like case based questions. Case based questions. Okay, so from this plasma membrane topic you are going to get the case based questions, and next you are also going to get the level 1 type of questions, that is section A, 1 marker questions, and next you can also get very short answer type of questions which are nearly 2 marks. Okay, so these three are the different type of questions which you can cover from the topic plasma membrane. Okay, and next coming towards the cell organelles. Yes, coming towards the cell organelles, we have different types of cell organelles and you need to study some important cell organelles which are important for the examination point of view. Those are nucleus. Clear, children? Nucleus, you need to focus mostly on the structure, function as well as its diagram. Diagram, its functions and its theoretical part. Okay, and next coming towards the endoplasmic reticulum, Golgi apparatus, ribosomes, lysosomes, plastids, vacuoles, and along with this one more. These are all the most important topics which we are going to discuss in this particular chapter. So without any delay, let's get started with the session. Yes. Okay. So cell, the fundamental unit of life. Okay. So actually what is a cell? What is a cell and why we are calling the cell as a structural and the functional unit of life? So let me break down this term and let me explain it in a clear way. So children, students here, each and every living microorganism from the lowest level of organization, from the lowest level of organization to the highest level of organization like chordates. Yes, from the lowest level of organization means, yes, we can take the example of sponges. Okay, from the lowest level of organization like sponges to the highest level of organization like chordates. Okay, so all the organisms from the lowest level to the highest level of organization, each and every living organism is made up of cell. The cells are tiny microscopic units. These are very tiny microscopic structures which are going to give the structure to the particular organism. We are going to call the particular organism as a living. Why we are going to call the organism as a living? Because it should possess the cell. When the organism is going to contain the cell, then only we can call that organism as a living organism. Clear with the definition? Cell is called as the structural and functional unit of life, and each and every organism is made up of the cells. Okay, if we look into the number, okay, based on the number, cells are divided into two types. Okay, so cells are divided into two types, unicellular and next one is multicellular. Clear children? Unicellular microorganisms are going to be present as well as multicellular organisms are also going to be present. So, in the unicellular organisms like amoeba, yes, in the unicellular organisms like amoeba, yes. So, in the unicellular organisms like amoeba, we can find only a single cell. Amoeba, it is a protozoan, it is a microorganism, and along with this paramecium is also a unicellular organism. So, in these microorganisms only single cell is going to be present. Okay, only single cell is present, and that single cell is involved in doing many multiple activities and many multiple functions like, yes, that single cell is going to be involved in performing many different types of functions like what? Digestion, respiration, circulation, and next one excretion. Yes, in the unicellular organisms, single cell is involved in doing the multiple functions like digestion, okay, and next one is circulation, and next excretion. Clear? So, that single cell is only involved in doing all these multiple functions in the unicellular organisms like amoeba. Okay. So, coming towards the multicellular organisms, in the multicellular organisms, what happens means, in the multicellular organisms, the single cell, okay, multicellular means what children? What is mean by the term multicellular? It means it is made up of more than one cells. Clear? Yes, multicellular means having some millions to billions or many number of cells. Okay, in that particular case, what happens means, so all the cells are going to aggregate. All the cells are going to be aggregate, and all the cells will group together to form tissues. Why they are going to aggregate? They are going to aggregate to form the tissues. Okay. So, now all the tissues are going to be grouped, or else all the tissues are going to be come closer to form the organs. Okay, here group of cells are going to form the tissues, and group of tissues will form the organs like brain, eyes, heart, next kidney, liver, different types of organs. Later, all the organs are involved in making the organ system. Okay, like it may be nervous system, circulatory system, excretory system, and so on. Clear now? So, all these organ systems are involved in making a complex individual, making a complexity structure of the individual to form organism. Clear? Clear with this conceptual map? Okay. So, here group of cells are involved in aggregation to form the tissues. So, later all the tissues are going to be aggregated to form the organs like kidney, liver, heart, and etc. And next group of organs are involved in making the organ, next group of organs are involved in making the organ systems, that may be either circulatory system, respiratory system, digestive system, and so on. Okay, and next that organ systems are involved in making a complete individual, that is called as the organism. So, clear with this concept? So, students, so we can observe all these things which are going to take place in the only multicellular organisms. Okay, in the multicellular organisms only the cells will aggregate to form the organism. Okay, but in the unicellular organism, when we have seen in amoeba, there only single cell is present, and that cell is only involved in doing the multiple functions like excretion, digestion, and next one circulation and everything. So, now whatever the activities we are going to perform in our daily life, okay, whatever the activities we are doing, suppose if I am writing on the board, if I am speaking, everything, these all are the activities, these are just the outcome of the cells. Okay, all the activities are just simply the outcome of the cells. We are going to do many number of multiple functions just due to the presence of the cells. Okay, now let's look into this definition. The cell is a structural and functional unit of every living organism. Okay, so in the unicellular organism it is doing many activities, hence it is called as a functional unit. But when it comes to the multicellular organism, it is involved in giving a proper structure to the individual by making the tissues and organs each and everything, hence it is called as a structural unit. So hence cell is called as a structural and functional unit of each and every living organism, and all the activities whatever we are doing, those are just simply the outcome of the cells. Clear with this concept, clear with the definition cell, students? Yes. And each and every cell is made up of the cell wall, and the cell wall is present in the plants, cell wall is present in the fungi, as well as cell wall is present in the bacteria. We are going to look briefly about the cell wall in the next slide. Yes. Yes. Here comes the discoveries of cell. Clear, children? Yes. Discoveries of cells. So in the discoveries of cells, you need to remember about the theories proposed by the different scientists, and you need to learn in detail about the cell theory. That is very, very important for your examination point of view. So yes, let's look into these discoveries of cell. So first, the scientist Robert Hooke is the one. Robert Hooke is the first scientist. He discovered the cell in the year 1665. Robert Hooke is a scientist. He has discovered the cell in the year when students, in the year 1665. So how he has discovered the cell? So he has observed the piece of cork. Okay, he studied the cell structure by taking the piece of cork under the microscope. Okay, so under the microscope he studied the structure of the cork. So when he studied the structure of the cork under the microscope, he observed the structure in this manner. So this is the microscopic structure observed by the scientist Robert Hooke. So when he observed the structure in the microscope, he observed some organization patterns like honeycomb. Clear, children? So he observed the honeycomb like structures which are present in the cork when he has seen under the microscope. So clear, this was the actual structure which is seen by the scientist Robert Hooke in his self-designed microscope. So when he has observed this structure, then it is proved like these are called as the dead cells. So he has concluded that these are the dead cells, because here we cannot observe any movement in these cells, and here we cannot observe any living characteristics in this particular microscopic structure. So this is the first cell which is discovered by the Robert Hooke. He discovered the cell in the year in the year 1665. So he studied what he studied, students? He studied the piece of cork under the microscope. So when he studied the cork structure, he observed the structure in the pattern called honeycomb structures. Clear? Yes. So he observed the honeycomb patterns under the microscope for the cork structure. Yes. So next, yes, based on this, one more scientist, Anthony van Leeuwenhoek. Anton van Leeuwenhoek, he discovered the cell in the year 1674. Anton van Leeuwenhoek has discovered the cells in the year 1674. He discovered the live cells. He was the first scientist to observe and describe the free living cells. So he observed the free living cells, free living different types of cells he has observed. Those are bacteria, and next one RBCs, okay, and next amoeba, and next one sperm cell. Clear? So Anton van Leeuwenhoek, he is the first scientist. He observed the living cells in the pond water. So he observed the bacteria, and next he observed the RBC. These are the RBC cells present in the humans, and next he observed the protozoans like amoeba. This is a unicellular one, and next he observed the sperm cells. Okay, it is a sperm cell which is involved in the reproduction. So he is the first scientist to observe all these living cells, whereas the Robert Hooke observed the cork structure, that was a dead cell. Okay, clear with the concept? Yes. Next, coming towards the two more important scientists, those are Robert Brown and Purkinje. Yes, Robert Brown. So Robert Brown is a scientist, he discovered the nucleus. So remember children, this is very important for your multiple choice of questions. Okay, in the 1 mark this question is going to come, so you have to focus on it. Okay, so Robert Brown is a scientist, he discovered the nucleus in the year 1831. He discovered the nucleus in the year 1831. He discovered the nucleus in roots of orchid plant. He discovered the roots, he discovered the nucleus where students, in the orchid plant. Okay, in the year 1831, it is very important. Who discovered the nucleus? Robert Brown discovered the nucleus in the year 1831 in the roots of orchid plant. Okay, yes. Next one, Purkinje. He is a scientist, he discovered the protoplasm. Okay, he discovered the protoplasm in the year 1838. So the protoplasm is a living content which is present in the cell along with the cytoplasm and nucleus. We are going to learn about this protoplasm in detail in the upcoming slide. Okay. Yes. Okay. Next one, these are two more most important scientists, Matthias Sliden and Theodor Swan. Okay, Matthias Sliden and Theodor Swan. So Sliden is a scientist, he is a German botanist. Okay, in the year 1838, he has proven that all the plants are made up of cells. So he has given the statement called cell is the structural and functional unit of life in all the plants. Cell is the structural and functional unit of all the plants. Okay, he has given the statement only regarding the plants. He is a Sliden in the year 1838. So he has given a statement called as the all the plants are made up of the cells. Okay, those are only the fundamental unit for those plants. Okay, and he is a German botanist. Okay, it is also very important. Yes, and next one, Theodor Swan. He is a German zoologist. Okay, so he has proven that cell is a structural and functional unit for all the animals. Okay, he is a scientist who has given the statement only regarding the plants. Okay, he is a scientist who has given the statement regarding only the animals. Okay, Sliden and Swan. In the year 1838, Sliden has proven all the plants are made up of cells. In the year 1839, Swan has proven all the animals are made up of the cells. Yes, clear children? Yes, now here, yes, Rudolf Virchow. Okay, so Rudolf Virchow, he is a German physiologist. So he actually proposed the theory of lineage. So actually what this Rudolf Virchow scientist says in the sense, so actually his statement is every cell is going to arise from the pre-existing cells. Clear with the concept children? If the cell is arised, that might be always, that is always going to be associated and that is always going to be arised from the pre-existing cells. No cell is going to be arised individually, it always going to arise from the pre-existing cells, and these two cells are arise from the pre-existing cells, and these are the cells again which are going to arise from these pre-existing cells. So in this manner, each and every cell is going to arise from those pre-existing cells only. Okay, this statement is given by whom? This statement was proven and it is given by the scientist Rudolf Virchow in the year 1855. He is a German physiologist. So based on this theory, that is every cell is going to arise from the pre-existing cell, he has given a great statement that omnis cellula e cellula. Okay, omnis cellula e cellula, it is only means for, this is only meant for the this concept children. Every cell will arise from the pre-existing cells, that is called as omnicellular, e-cellular. It means, see here, new cells are going to arise from the pre-existing cells. Clear with this concept? Yes. These theories and discoveries regarding the cells are very important for your multiple choice of questions children. So, please try to memorize each and everything. Yes. Next, coming towards the two most important scientists, those are Knoll and Ruska. So, these both are the scientists in the year 1932. These are, both of them have designed the electron microscopes to study the ultrastructure of the cells. So, generally students, if you see, the cells are invisible to our naked eyes. Those are very tiny microscopic structures. We cannot study them by with our naked eyes. Okay. So, those are invisible. So, that is the reason which gave these both scientists Knoll and Ruska to invent the electron microscope. Okay. With the help of this electron microscope, we can study the fine structures of the cells, and we can study in detail the structure and its composition in detail by the by using the electron microscope children. So, now we will study about the microscopes. Okay. So, we have seen all the discoveries of cells which was given by the different types of scientists. Okay. So, we have seen that Robert Hooke is the scientist who discovered the cell, and he observed the cell structure by placing a piece of cork under the microscope. And next, we have also studied the Purkinje. He is a scientist who has given the term and who has discovered the protoplasm. And next, Rudolf Virchow. He was a scientist who has given the statement omnicellular and e-cellular. And next, Antony von Leeuwenhoek was the scientist who has observed the living cells. And next, we have also seen the two most important scientists. Those are Sliden and Swan, who has proposed the theories that all the animals are made up of cells, and all the animals are also plants and animals are also made up of cells. So, based on all these cell theories, by studying each and all these theories, the final conclusion is been given, which is going to include three most important points. Okay. The cell theory is modified and it is stated as follows. Yes. The cell theory is modified and it is stated as all the organisms are composed of the cells and the products of the cells. And here next one is new cells are always going to arise from the pre-existing cells. Already we have discussed about it just now. Okay. Who proposed that? Omnicellular e-cellular process. Okay. Rudolf Virchow. Okay. Don't try to forget children. You have to remember that, that is very important. Omnicellular e-cellular was given by Rudolf Virchow. Okay. And that statement is going to state all the cells are going to arise from the pre-existing cells. Okay. And cell is generally called as the structural and functional unit of the life. If we are calling it as a living, then it should possess a cell undoubtedly. Okay. Yes. Now, here comes the most interesting one, that is microscope. Yes. Okay. Light microscope. So, what are microscopes actually? What are microscopes, students? So, microscopes are the instruments. Okay. Microscopes are the instruments. We use the microscopes generally to see the cells. Okay. Generally to observe the fine structure of the cells. Okay. Generally the cells are very microscopic tiny structures. We cannot see them nakedly, and we cannot study the entire structure by looking. Clear, right? So, these are the microscopes. These are actually the instruments which are used to study the fine structure and the detailed structure of each and every cells. Okay. So, this is the light microscope. So, now we are going to study about the light microscopes, and then next we will study about the electron microscope. Who invented the electron microscope? Electron microscope was invented by Knoll and Ruska. Okay. So, this is generally a light microscope. This is also called as compound microscope. Okay. Compound microscope. Okay. And next, it is also called as optical microscope. Okay. Light microscope is also called as compound microscope. It is also called as the optical microscopes. So, okay. So, in our daily life, in our laboratories, whatever we are using the microscopes, those are mostly light microscopes only, children. In your school activities or else in the lab activities, whatever we are going to use the instrument now, that is the light microscope. Okay. This is a most commonly used microscope. So, now we will study in detail about the parts and its functions. Yes. So, this light microscope is going to contain some parts. Okay. This microscope is going to contain some parts. First, it is going to contain one arm. What it is called as arm. Okay. So, the arm is just a handle. Okay. The arm is just to hold the microscope. Okay. And next, this is the base. Okay. This will be the base of the microscope, and this is the arm of the microscope. Okay. This is the base where all the parts and total microscope is organized in a proper way. Okay. And next, here we have a stage. Okay. The stage is a place where we have to place the specimen, and where after preparation of the slide, we have to place the slide along with the specimen on the stage. Okay. On the stage, you have to place the glass slide along with the specimen. After placing the slide, to hold that slide in a proper manner, there here we have the special structures called as clips, objective clips. Okay. These are the clips. Okay. That clips, what they are going to do? They are going to hold the slide in a particular manner. Okay. So that you can see the cell structure clearly. Okay. The stage clips are going to hold the slide. And next, we have here objective lenses. Okay. These objective lenses are going to be present in 3 different types of powers, like 10x, yes, 10x, okay, 5x and 15x. Okay. So, these are the objective lenses, these 3. Okay. These 3 are the objective lenses with different types of magnification powers, and here we have the light source. Why we are calling it as a light microscope, students? Because this microscope is going to use the light as a source of illumination to view the cell in better way. Okay. So, this is the light source. From here it is going to emit the light, and this light is going to pass to this specimen present in this slide, and we have to look this specimen in detail from the eyepiece. Clear? Yes. From the eyepiece we can clearly see this specimen which is present in the slide, which is placed on the stage. Okay. So, here this will be the light source. It is actually a condenser and it is a mirror. Okay. It is going to use the sunlight. Okay. And from here all the light is going to be passed. Okay. And all this light will pass and it will reach to the specimen, and when we look it from the eyepiece, we can get the clear image of that particular cell structure. Yes. And next, here we have the revolving nose piece. Okay. You can adjust all those magnification powers as per your preference, that is based on the cell type. Okay. And next, here we have the fine adjustment and coarse adjustment knobs. These are just meant for the proper visualization. You can either increase those adjustment knobs, you can or else you can decrease those adjustment knobs to have the proper vision of that particular specimen. Clear with the light microscope, students? Light microscope is an instrument which is most commonly used in all the laboratories, and we use this particular microscope to see the cells which are invisible to our naked eyes, and we can study the proper structure and we can clearly observe the fine details of the cells. So, and this microscope is going to be also called as the compound microscope, or else it is also called as the optical microscope. Why we are calling it as optical microscope means, here we have the condenser lens that is made up of, that is a concave lens, and from this lens the light is going to emit. Okay, the light is going to emit, it uses the light as a source of energy, hence we call it as the optical microscope, and hence we call it as the light microscope. Okay. and from here the rays will pass and it will reach to the specimen which is present on the slide and which is placed on the stage, okay, and we can get the clear vision of the specimen when we are going to see it from the eyepiece, okay, and it is going to contain the arm to hold the microscope, and it is a base where all these parts are going to be constructed in a proper way, okay, and this will be the stage and these are the stage clips to hold the slide in a proper position, okay, clear with the light microscope? Yes. So, next moving towards the electron microscope. Mostly you are going to get the questions from the light microscope only, but it will be better if you study about the electron microscope also. Yes. So, in the electron microscope, what we are going to use as a source of illumination? In the light microscope, we have used the light as a source of illumination. In the electron microscope, we use electrons. We use the electrons as the source of illumination to see the specimen. Clear, children? So, here the electrons will pass, okay, the electrons will pass and the electrons are going to reach the specimen, and that image is going to be identified by the electromagnets. And next one, the clear image we can see here, okay, the clear image is going to appear in the fluorescent screen, okay, fluorescent screen, okay. The electrons are going to eject, and then next that picture is detected by the when the picture is going to be detected in the presence of electromagnets, and the clear picture, the clear structure of that cell or else the clear structure of that particular microorganism or whatever, that is going to be appeared on the fluorescent screen. Yes, this one, and from here the process is going to start, and here we can get the clear structure. Yes, this is all about the electron microscope. Yes. Students, now here comes the cells based on the composition. Cell based on the composition, okay. So, cell, here we need to study about the cell based on the number, cells based on the size, cells based on the shape, okay. Number, size and shape. Clear? These all three things we need to study in the composition of the cell. So, now let us look into the based on the number. Cells based on the number are going to be divided into two types. One is unicellular and another one is multicellular. Clear? So, in unicellular organisms, what we are going to have, children? We are going to have only single cell. So, these organisms are also called as protozoans. Protozoans are the microorganisms which will possess only single cell, okay. And next, multicellular organisms are made up of more than one cell, okay. So, based on number, here these are the examples, okay. We have amoeba, okay, it is a unicellular, and next euglena, okay. Euglena is it is considered as both the animal as well as the plant, it is a protist. And next one, Chlamydomonas, it is a unicellular algae, and next one Paramecium, it is also a protozoan. Yes, coming towards the multicellular organisms, yes. So, here cells will aggregate to form the tissues, and then it will aggregate to form the organs, and next organ systems, and the next one is individual. These already we have seen in the first starting only, okay. And next, these are the plants. Here cells will aggregate to form the tissues, and next it is going to form the organs like roots, branches, stems, and next completely later it is going to form the complex structure called as organism, okay. Here it will form the plant, here it is going to form the individual, single individual, that may be either humans, animals, or whatever it may be, okay. Yes. Next, cells based on the shape, okay. Based on, this is very, very important in the examination point of view. From here you can get the 1 mark questions, children. So, please try to understand this concept, okay. So, cells based on the shape, and here cells based on the size. So, first we will look into the cells based on the size. So, based on the size, the smallest cell, it is very, very important, okay. It is very, very important. The smallest cell is mycoplasma. The smallest cell is mycoplasma, which size is going to range from 0.5 to 0.1 to 0.5 micrometers. Okay, and these are the only microorganisms which are not going to have any cell wall. Okay, these mycoplasmas are also called as PPLOs. Okay, PPLOs are nothing but pleuropneumonia like organisms. These are the very smallest microorganisms which are not going to contain the cell wall, and their size is going to range from the 0.1 to 0.5 micrometers. And the largest cell is cell of an ostrich. Okay, cell of an ostrich. Yes, here if we look into the size, it is going to range from 15 centimeters in length and 13 centimeters in width. These two are very important. Smallest size, smallest cell is a mycoplasma, and largest cell is the egg of an ostrich. Okay, and its size is going to vary from 15 centimeters in length to the 13 centimeters in width. Okay, and here in the mycoplasma the size is going to vary from the 0.1 to 0.5 micrometers. Yes, now we have to look into the cells based on the shape. Cells based on the size has been completed, and next cells based on the number also we have studied. Now we are going to look into the cells based on the shape. Okay, see children, cells are not always going to be in a circular manner. Okay, the cells are not always going to be in a circular manner. Okay, the size and the shape of the cell is going to vary in different organisms. Okay, so now if we look here, these are the RBCs. Okay, these are the red blood cells of the humans. Okay, these are biconcave in structure, and its size is going to range from 7 micrometers. Okay, its size is going to range from the 7 micrometers. These are the RBCs which are biconcave in structure. And next, these are the fat cells. Okay, these are fat cells. These are the specialized cells. These are generally specialized to store the fats, and this shape is globular. Okay, these are biconcave, these are globular in structure, and it is a smooth muscle cell. It is a smooth muscle cell, and this smooth muscle cell is going to give the structure like spindle shape. Okay, the smooth muscle is in a spindle shape. Spindle shape means it will be in this structure. Okay, it is going to be broad in middle and pointed at the ends. Okay, this is a smooth muscle structure which is a spindle shape. Okay, and next one, these are the bone cells, and next these are the sperm cells and the ovum cells. Okay, it is a sperm and ova. These are also cells which are involved in the reproduction process, and we also have the white blood cells which are going to show the amoeboid shaped structures. Okay, this is all about the composition of the cell based on the number, size, and the next one is shape. Okay, these two are very important. Please just note down, students. Yes, next, here comes the structure of the prokaryotic cell. Yes, structure of prokaryotic cell. Yes, students, this topic is very important. Its diagram and its functions is very important. So please everyone get ready with your notes and please note down the points. Yes, structure of the prokaryotic cell. Yes, so before studying about the prokaryotic cell, I just want to ask you all the students, what is eukaryotic cell? Okay, so already all of you have the basic idea about this chapter, because already you all have studied it in the 8th standard, children. In the 8th standard, this is the last chapter, cell, the fundamental unit of life. It is a chapter already included in your 8th standard. So now you need to answer the you need to give the answer about the prokaryotic cell and the eukaryotic cell. Okay, so before learning about this complete prokaryotic cell structure, so let me make you a clear understanding about the eukaryotic cell. Yes. So what is eukaryotic cell? Okay. The differences between the prokaryotic and eukaryotic cell. This concept is very important, and these question you may get for the 5 marks or 4 marks, children. Okay, so listen it carefully. Yes, in the eukaryotic cell, in the eukaryotic cellular organisms, we are going to find some important membrane bound cell organelles, that may be at the single membrane bound cell organelles or double membrane bound cell organelles. In the eukaryotic cells, we are going to find the true nucleus. What we are going to find, students? We are going to look the true nucleus is present in the eukaryotic organisms. Yes, true nucleus is present, okay, in the eukaryotic organisms. So what is meant by true nucleus? So when we are calling the term, when we are giving the definition of the term the true nucleus, it means it is going to show some important characteristics like nuclear, yes, let me write here. So the term true nucleus is going to possess 4 important characteristics like nuclear envelope, and next one nucleolus, and next one nucleoplasm. Okay, these are the important characteristics which we can find only in the eukaryotic microorganisms. Okay, so in the eukaryotic organisms, we are going to find the genetic material. Okay, the genetic material, the DNA is going to be present, and that genetic material is covered with the nuclear envelope. This complete genetic material is covered with the nuclear envelope in the eukaryotic microorganisms. Okay, in the eukaryotic organisms, the presence of the nuclear envelope only, due to the presence of this nuclear envelope only, we are going to give the definition of the proper term true nucleus. Okay, true nucleus means it should contain the nuclear envelope along with the genetic material. Okay, so along with this, in these eukaryotic cell, we can find the single membrane cell organelles and double membrane bound cell organelles like endoplasmic reticulum. We are going to study about all the cell organelles in the next upcoming session. So now let's have a short look on each and every cell organelle. Okay, so we have the endoplasmic reticulum in the eukaryotic cell. It is involved in synthesis of lipids and proteins. And next one, Golgi body. Okay, and next here we have the yes, Golgi body is also a cell organelle which is involved in packaging, modification and secretion of proteins. Packaging, secretion and modification. Okay, this Golgi body is going to perform all these 3 activities. Okay, it will pack the proteins, it will modify the proteins, and then next it is going to synthesize the proteins. And next one is the mitochondria. As you all have the basic idea about it, mitochondria is called as a powerhouse of the cell because it is going to generate or else it is going to release the energy in the form of ATP. Okay, and next here we have the lysosomes. In the eukaryotic cell organelle only we are going to have all these organelles, children. Okay, don't get confused. Okay, in the prokaryotic cell, when we are going to learn about the prokaryotic cell, then you will get a proper identification how that actually going to differ from the eukaryotic cell. Okay, so next we have the lysosome. It is a single membrane bound cell organelle which is involved in breaking the cellular waste materials. Okay, and next we have the cell membrane. Okay, each and every cell is going to be covered by the cell wall along with the cell membrane. Okay, and next cytoplasm and the next ribosomes which are mainly involved in the synthesis of the proteins. So now clear what actually the eukaryotic cell is going to contain. Eukaryotic cell will contain the nucleus and next endoplasmic reticulum, Golgi body, mitochondria, lysosomes and chloroplast, many other things. Okay, so the main thing we have to focus is in the eukaryotic cell we have the true nucleus which will possess the important characteristics like nuclear envelope, nucleolus and the next nucleoplasm. Okay, here the genetic material is present and which is completely covered by the nuclear envelope. Okay, you have to remember this. So I am going to ask you the questions regarding this particular nucleus only, children. Okay, so when we study about the prokaryotic cell structure, you need to answer about the how it is going to differ from the eukaryotic cell. Yes. So this is the structure of a prokaryotic cell. Okay, in the prokaryotic cell, yes. So this is actually the structure of the prokaryotic cell. It is a typical bacterial cell. So here if you see, these are many bacteria. Okay, when they are many, then we call it as a bacteria, and when we are going to study about its single structure, then we are going to call it as the bacterium. Okay, so these are the microscopic structures of the bacteria. So now these bacteria is a single cellular, it is a unicellular. Okay, so now we have to study only about this single cellular structure of this prokaryote. So when we study about that particular single cell structure, then we can observe this complexity. Okay, then we are going to observe all these structures in that particular prokaryotic cell. So in the prokaryotic cell, you need to learn about the three most important structure, the glycocalyx, and next one cell wall, and the next one is cell membrane. Clear, children? So in the prokaryotic cell, you need to learn about all these three things. Actually in your NCERT book, okay, in your NCERT standard book, only the bacterial cell structure along with the functions has been given. So in the next upcoming session, we are going to learn in detail about its parts, about each and every structure we are going to learn in detail, and it is very important for your next higher standards also, children. Okay, so please I request all of you, and I am going to give you a question that please everyone try to answer what is the difference between the prokaryotic and eukaryotic cell, and next, okay, and next in the next live session I need an answer from you students. Okay, so I will be waiting for all your answers. So please stay tuned, and please everyone, I request everyone to join in the next upcoming session. Thank you.